Hey everybody, it's your life science slash biology teacher, Mr. Poser. Today we are getting into our fifth unit, which is on the cell cycle. And our first topic on the cell cycle is going to be, well, the cell cycle. In our next video, we'll get into more detail about the phases of mitosis and how exactly cell division occurs. But I don't want to get too ahead of myself here because I don't want to give any of the answers away to these questions that I'm about to ask you here to start off this video. All right, so as always, I like to ask these questions, and hopefully you might have an idea of how this works before the end of the video. So here's a bunch of human tissues. You have some epithelial tissue over here that you might find in your intestinal lining. You have bone tissue over here, and you have blood down there. These are all different types of blood cells. And in fact, an amazing thing about the human body, or lots of different organisms, is that every 4 to 5 days-ish, you get a brand new intestinal lining. That means all the cells in your intestinal lining are brand new every 4 to 5 days. Same goes for the top layer of your skin called your epidermis, which you can see right here. That's replaced every 2 to 3 weeks. Your blood's replaced every 4 months-ish. And your skeleton, actually, all the cells in your skeleton are replaced every 6 to 8 months. And how is this possible? How can human body parts replace themselves? So that's a question I'm going to leave open-ended here for this video. Secondly, okay, I have a couple pictures here, and I want you to try to figure out what is happening in each of these images here. So it looks like we have a ball of cells over here with another cell kind of like, or a layer of cells kind of covering it, and then we have some black cells, some purplish blue, like dark cells that seem to be kind of getting into the bloodstream over here with these red blood cells. And what is this? What's happening in each of these images? And all of these tie together here over the course of the video. All right, so I'm going to bring this image back here just one more time here. So this is what we've seen a bunch of times. Here's our protocell. It's able to maintain an internal environment. It's able to maintain homeostasis, reproduce its RNA, metabolize, react to changes, and dispose of waste. And therefore, it's a living thing. And if we are prescribing to a well-known, well-established theory called cell theory, then we know that this protocell, well, for lack of a better word, it's a cell. By all definitions, it's able to do all of those things. So the last requirement of being a living thing is being made of at least one cell, and that's what cell theory states here. Cell theory states that all living things are made up of at least one cell, and all cells come from other cells, pre-existing cells. This is a novel concept back when it was first discussed, and people started looking at things right under the microscope for the first time. So, imagine that. Nobody could really think of any tiny, tiny microorganism, invisible microorganism, something that's too small that you can't even see it. It's a living thing. That's inconceivable until this guy down here and a couple others started looking at our microscopes. Antony van Leeuwenhoek and Robert Hooke were some of the first ones that looked at these little tiny microorganisms that make up our own bodies and are their own microorganisms themselves, called cells, and helped develop cell theory. So multicellular organisms, even like human beings, develop from just one cell, though. All of us at one time in our lives were just one cell. And cells, just like human beings or any other multicellular organisms, they are born, they grow, they reproduce, and they die. And they go through a life cycle just like any other organism might. Okay? So, by the way, fun fact, the reason why they're called cells is because it was either this guy or another one thought that, these cells, these are onion cells, and which are representative of what they might have looked at underneath some of the world's first microscopes. He thought they looked like rooms, or rooms in a monastery, which are called cells. So that's why we call cells cells, interestingly enough, because he thought they looked like rooms in a monastery. But anyway, they go through a life cycle, and they reproduce just like any other multicellular organism. Except, what do cells do to reproduce? Well, they divide, and what we're going to see here is this is a diagram of what we call the cell cycle, the life cycle of a cell, the life and reproductive cycle of a cell. And we're going to talk about each of these phases here momentarily. And so I'm going to go ahead and flip to the page. And here's what the cell cycle is. It's the regular pattern of growth, DNA duplication, and cell division that occurs in cells with nuclei. So we are dealing with eukaryotes here. Prokaryotes have a slightly different cycle, but we're going to be focusing on eukaryotes today. And we can break down the cell cycle into 5 main phases here. Those are G1, S, G2, M, and another one called G0, which we'll add on the next page here. Okay. So in each phase, so if this were about 24 hours, some cell cycles take 24 hours, so we can use that as kind of a frame of reference for talking about how long each cell or a cell stays in each phase here. So G1, this is the first phase here, and this is where a cell grows. It carries out its normal functions. It does cell things. It does what cells do. It makes proteins. It does its job. Whatever. Okay, and that's the normal phase. It's called G1 or gap 1, as it's often referred to. And this is a decent amount of the cell's life here is spent in gap 1. Maybe if this were 24 hours, it would spend about 8 of those hours in gap 1, just doing normal cell stuff. Okay, the phase following that, okay, if the cell passes what's called a checkpoint and makes sure everything's good before moving to the next phase, it's called S, or the S phase, and or S stands for synthesis. And this is the stage where a cell makes a copy of its DNA. Now, why would it need to make a copy of its DNA? It needs to make a duplicate of all of its genetic information. If you remember from way back from Unit 1, DNA and RNA are a cell's instructions to make proteins. They're the blueprint for how to build a living thing, essentially. So in order to build another living thing, in order to reproduce and actually turn from one cell into 2 cells, you need to be able to copy the DNA so that each cell gets an exact copy. And you know that's what cell division is. It's like cloning, and you're getting a brand new copy of, but it's the same copy. You're getting a brand new copy of DNA for every new cell that's formed. Okay, so it needs to be doubled or duplicated or replicated. There's lots of ways to put it. But that happens in S phase. So then following S phase, we enter G2, which is gap 2. And this is where the cell grows a little bit more, and it prepares the cell for division. It gets ready to divide and split into 2. So imagine that. If you were to try to clone a human being, how much preparation would that take? That would take a really, really long time. So the cell has to make sure everything's right in order for it to split and make a copy of itself. Okay, and then M stands for mitosis. This is the stage in which the cell divides. It actually takes time to make an exact copy of itself. One cell becomes 2 exactly the same identical cells. All right, so those are the 4 main phases of the cell cycle when cells decide to divide. And an important point here is that different types of cells divide at different rates depending on the growth or the function of the cells. Okay, so remember how cells are differentiated. They have different jobs from one another, and they do different tasks within a multicellular organism. Sometimes you're going to need intestinal lining cells every 4 to 5 days, right? So cells are going to be dividing much more in there than they say would in a muscle or in a bone, for example. Cells are going to be dividing very, very fast. Or on your skin, for example, your epidermal cells are going to be dividing much faster. Okay? But some cells don't divide at all, and that's our 5th phase of the cell cycle, is that some cells don't divide and they stay in what's called G0. They carry out the functions, but they don't divide unless they receive a signal saying, hey, you have to divide. Okay, so some examples like neurons, you have about a couple trillion of them in your brain. They do not replace themselves at all. Okay, they are amitotic, meaning that they will never divide and they will stay in this G0 phase for the rest of their life. Okay, so that's G0 phase, and those are the 5 different phases of the cell cycle. Okay, so cells have to divide. They have to split into 2 in order to keep themselves going. To keep if you're a type of bacteria, you want to keep the colony going. You have to keep splitting and splitting and splitting, because nothing lasts. Well, nothing lasts forever. And, well, they can't just keep growing in size forever. Okay? Cells can't just keep going and getting bigger and bigger and bigger and bigger. Yeah, they grow, but they can't get that big. And we're going to talk about why that is, what limits cell growth here. And there's a short answer to this that we're going to get to on the next page. But cell growth and division are regulated by several factors here. Cells can't keep growing forever, but they also can't just keep dividing forever. Okay, so there's limits to, and there's regulations to the growth and division of a cell. Okay, so cells are limited in their growth because of what's called their surface area to volume ratio. Okay, so now, your surface area to volume ratio here, This is something that you might have calculated maybe in math in elementary school or maybe in middle school. But to calculate the surface area of a cube, right, you're going to go multiply the side length time. You're going to square it and then multiply it by 6 to find out the surface area of the cube. Okay, and you divide that by the volume of the cube, which would be 1 cubed, 1 times 1 times 1 if it were 1 centimeter, and then you get a ratio. Okay, the higher the surface area to volume rate, or excuse me, the lower the surface area to volume ratio, the worse it is for a cell. Okay, so as you can see, as this cell gets bigger and bigger and bigger, that becomes more of a problem. The surface area to volume ratio is getting smaller and smaller and smaller. And why that is, the really important part of that, if you were in AP Bio, it'd make you calculate that and figure out, which cell is better at absorbing and releasing materials and exchanging with the environment. But for this class, okay, I'm just going to have you know that cells depend on diffusion of nutrients and waste, not things kind of naturally moving in and out. And the reason why they can't keep growing forever is because if they get too big, they can't release and absorb materials quickly enough because their surface area is not large enough for their volume. Okay? Their surface area to volume is too small, and they can't grow in size forever. That's why you don't see, if I were a 6-foot cell, that wouldn't make any sense at all, because I would have to wait for oxygen and carbon dioxide to kind of just absorb into my body naturally and wait for it to absorb and diffuse naturally back out, which happens way, 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 way too slowly. Okay? So that's why you can't just be one ginormous cell, and that's what limits cell growth. Cell division is regulated by what we call checkpoints. Okay? They can't just grow out of control, or they shouldn't just grow out of control. Sometimes they do, and we're going to talk about that in just a second. But they're throughout the cell cycle. Okay? This is another depiction of the cell cycle over here. The cell has to pass through what's called checkpoints, which are internal factors and external factors that make sure that, hey, can we move to the next phase of the cell cycle? Can we actually divide safely? Are we in the proper condition to divide? Is the cell large enough? Is the DNA copied in the right way? These are checkpoints that the cell has to pass in order to make sure, like, okay, you're cleared to divide. Okay? And it needs to be regulated. It can't just keep dividing and dividing and dividing and dividing. So these things called checkpoints, which are, yes, like I said, internal factors that make sure the cell's dividing properly, make sure that the cell does not make any mistakes as it's dividing, or if it doesn't divide too much. Okay? And when cells divide too much, that's a problem. Okay? And if the cell is really, really out of control, one way that cell cycles can be regulated is through, if you really just press the panic button here, and you just, cut it off, and you trigger what's called apoptosis. Okay, so think about this. You have some cells in your body, and they're dividing, but there's something really wrong with them, and they're dividing and dividing and dividing and dividing and dividing and dividing, and they shouldn't be dividing as much as they are, or they have some kind of serious problem with them. What your body might be able to do is send those cells a signal which will cause them to self-destruct, which just basically explode. Well, I don't want to say explode. Things don't explode in biology, just so you know, but I take that back. But it'll break apart, and that's called apoptosis, which is one of the craziest concepts in biology, if you ask me. And apoptosis is defined as programmed cell death that occurs when signals cause a cell to produce self-destruction of enzymes. So it begs the question, why would you want your cells to die? Well, if the cells are really out of control, or if they're really damaged, or if they're harmful to other cells, then we're going to have to send those signals to make those cells self-destruct, which is a really interesting concept. But yeah, apoptosis, that's one of the ways that your body is going to try and fight against uncontrolled cell division, which is also known as cancer. Cancer is the result of unregulated cell growth. So when cells are going through the cell cycle, and they're just kind of dividing and dividing and nonstop, recklessly and very quickly, that's called cancer. And that's when your own cells divide uncontrollably. They kind of skip the checkpoints, and they just say, oh, just divide, just do it, just go, just divide. There's no regulation, and there's no control when it comes to cell division here. So human cells become cancerous when they can no longer regulate. They don't care about the checkpoints anymore. They can no longer regulate their own division, and it stems from abnormal or mutated genes, which is going to be a huge topic in our next unit. Genes are. So this is what this is some artist depiction of what cancer looks like. Some cells that are abnormal, or they kind of just forego all the regulations of the cell cycle. They just keep dividing and dividing and dividing, and that's when they form what's called a tumor. And if the tumor gets large enough, what cancer is really good at doing is entering the bloodstream. So if you've ever heard of cancer becoming or metastasizing, or lung cancer say become brain cancer or bladder cancer or colon cancer or anything like that, so what something that cancer is really good at is getting into the bloodstream and spreading out throughout the body. So when the cell is uncontrolled dividing, that is called cancer. Okay, so let's get back to these two questions here. How do your body parts replace themselves? Well, the short answer is through cell division. Intestinal and skin cells make a lot of physical contact. The cells basically replace themselves. A lot of your skin, you lose pounds and pounds of your skin every single year. It just kind of sloughs off or gets brushed off, and it becomes the dust in your house. But those dead cells that are brushed off of your body are replaced by new cells that come from underneath. And how are they replaced? Well, it's because of cell division. They're making new copies. Blood cells have a short lifespan, and they have to reproduce quickly as well. So all your blood cells get replaced throughout your life, but all of your blood cells will be replaced in a 4-month time period. And same with bone. Actually, bone cells are constantly dissolving old brittle tissue and replacing it with new tissue. So that's how you get a new skeleton as well. All right, so last two things here. What are happening in these images? Well, I think we've already figured out this one, that this is a representation of cancer. But what's happening here? Well, this is the shape of, this is what's called a blastula. It's a fertilized egg. So when sperm and egg combine after fertilization, it forms what's called a zygote, and that's the first cell that's going to be making up a multicellular organism like yourself. So we were all just one cell called a zygote. It forms a ball of cells, and it implants into the uterine wall, into the uterus of the mother. Okay? And then it forms a cup shape, and I'm going to challenge you to guess what human body structure the opening of the cup shape becomes. It's called it's a blastocyst it becomes, and I want you to figure out. I'm just going to let you figure that out for yourself. Maybe just do a Google search. All right. But anyway, one cell becomes this ball of cells, and that ball of cells eventually will become specialized, and it will become a fully functional human being. Hey, and then last one, of course, this is cancer. Cancer cells invading a bloodstream, it can travel to other parts through the blood and get to any other part of the body. So this is their uncontrolled cell growth. And entering the bloodstream is called metastasis. All right, that's it for this video. Please let me know if you have any questions. We'll see you next time.